Chapter 6, Nutrition in Humans. The definition of nutrition, the meaning of nutrition, is organisms obtain food. The definition of food are materials that can provide energy to the organism, materials that can allow the organism to grow and repair its body tissue, and also materials that can maintain health. There are different ways of nutrition. We call them mode of nutrition. What is the mode of nutrition in humans? And that is what we are going to look into right now. Organisms can be classified into two major groups according to their mode of nutrition. 1. Autotrophs. 2. Heterotrophs. For autotrophs, organisms can synthesize or make organic nutrients from simple inorganic substances. For example, plants can do something like this. Plants can convert inorganic materials, that is carbon dioxide, CO2, and water, H2O, to organic nutrients, that is glucose, which has the chemical formula of C6H12O6. The word organic nutrients refer to materials that consist of carbon, with an exception of carbon dioxide. Autotrophs refer to the organism itself. The mode of nutrition that the autotrophs practice, the autotroph perform, is called autotrophic nutrition. Heterotrophs cannot synthesize organic nutrients. They must obtain nutrients from other organisms or dead organic matters. For example, animal, bacteria, or fungi. I have some notes for logical thinking here. Logical thinking involves the gathering of a number of facts and then give deduction. These facts can be definition, can be things that you observe. Human takes in organic food. It's something that you can observe. Cannot synthesize or make organic nutrients is also something that you can observe. When matching with the definition of autotrophs and heterotrophs above, we will be able to deduce that humans are heterotrophs. This way of thinking is called logical thinking, based on accurate facts and observation. We make deduction. Within the category of heterotrophic nutrition, there are three types: holozoic, saprophytic, and parasitic nutrition. Which type of heterotrophic nutrition does humans belongs to? There is some logical thinking here again. We have to base on the definition again. Let's look at holozoic nutrition. Holozoic nutrition refers to the type of nutrition where animals take in complex organic compounds in form of solid food particles. And also, in this type of nutrition, food is digested internally into soluble product that is small, therefore it is soluble products which are then absorbed. For example, absorbed into the blood. Organisms that practice this kind of nutrition we call holozoic nutrition, can be animals. Saprophytic nutrition. Saprophytic nutrition refers to a kind of nutrition where organisms obtain soluble organic nutrients from dead organic matters. Another fact is that this kind of nutrition have food that is digested outside the body before absorption. Example of organism or living things that practice this kind of nutrition we call saprophytic nutrition can be bacteria and fungi. Please stop the video now and type these two words, bacteria, fungi, into Google to find how they look like. Let us continue. The last of the three types of heterotrophic nutrition is called parasitic nutrition. Parasitic nutrition refers to organisms that can be able to obtain organic nutrients from other organisms. We call this other organism the host, and the organism that obtain organic nutrients like this is called a parasite. The food obtained by the parasites may or may not need digestion. Examples of living things that practice parasitic nutrition are bacteria, fungi, protists, and worms. And I would like you to focus on going to the Google web and type the word protist and another word called malaria, where malaria is the name of a disease where a parasite inhabit, take shelter, or live in your red blood cell. It is really fascinating. Please stop this video and have a look. Just type the word malaria and the word parasites in Google. In summary, humans, by definition, practice heterotrophic nutrition, particularly holozoic nutrition. 2. Nutrition in human. In this section, we focus on the processes that is involved in nutrition in human. The first process is called ingestion. In ingestion, we focus on learning the structure of human tooth. The basic structure of human tooth is separated into enamel, dentine, and pulp cavity. Enamel happens to be the outermost layer of the teeth, which cover the crown only. It is non-living. That means it doesn't have living cells. It is very hard and resistant to decay and wearing. It is made of calcium, has very little organic matter such as carbohydrates, protein, and fats, very little of these organic matters. Dentine is the middle layer of the teeth. It is considered as living because there are strands of living cytoplasm running through the dentine. So living cytoplasm happens to be part of the cell, so we can consider dentine as a living part of the teeth. Dentine is softer than enamel, but it is still quite hard as bone. It is made of calcium with more organic matter, which we just refer to as living cytoplasm, strands of cytoplasm that runs through the dentine. Pulp cavity is the innermost layer of the cell. It consists of living cells. There are blood vessels for supplying oxygen and nutrients. There are also parts of living nerve cells that is called nerve endings for detecting stimuli. These nerve endings, we usually have a special name for them as being pain receptor, thermal receptor, which is referring to heat, hot or cold receptors. Here we have a vertical section of a human molar, one kind of teeth. There are different kinds of teeth. Incisor, canine, premolar, and the largest one called molar. This is a vertical section of it, showing the outermost layer, the middle layer, and the inner layer clearly. So naturally, we'll move on to the different type of teeth that we possess. Incisor, it has sharp edge like a chisel. Its function is for biting and cutting food. Canine is pointed. Its function is for biting and tearing food. Premolar and molar has broad top with cusps. Its function is for crushing and grinding food. So sharp-edged, like a chisel, like this, looking at the front, and on the side, like this. So front view and side view. For canine, it is pointed, so its front view look like this, pointed. Its side view also look like this. So either from the front view or side view, it's pointed in shape. Premolar, broad top with cusps. These are cusps. Next, we move on to human dentition. There are two kinds of human dentition. Number one is the milk teeth. The second one is the permanent teeth. The total number of teeth for milk teeth is 20, where there are no molars. The permanent teeth, the total number would be 32 for adult and 28 for teenager, because the last four are what we call the wisdom teeth, which may never come out in adult, but if it comes out at the adult age. For milk teeth, they are replaced by permanent teeth. For permanent teeth, the fact is that once they are lost or decayed, they cannot be replaced. Regarding the process of ingestion, we have to discuss swallowing. In swallowing, the larynx is raised and the epiglottis lowers to cover the trachea. So that the bolus will enter the esophagus. The importance of the epiglottis covering the trachea is to prevent food from entering the trachea. Digestion is the next process of nutrition in human. Here is a general plan of the alimentary canal and its glands. You can see glands such as the pancreas, you can see glands that is, for example, liver. There are glands also on the walls of the ileum. There are glands also on the wall of the duodenum, that is the small intestine. The alimentary canal is basically a tube with different parts of the canal of the tube differentiated, changing shape into, for example, the stomach, the mouth cavity, the ileum, the duodenum, which serve different functions. The first part of the alimentary canal is called the mouth cavity. It is responsible for mechanical digestion, a kind of physical digestion which involves chewing of food. Salivary glands there are in the mouth cavity to secrete saliva, which contains salivary amylase to convert starch to maltose. This is the general macro structure of starch, and this is the general structure of maltose, which consists of two glucose. Starch, which consists of many glucose linking up together. When they break down, you should know that it is a kind of reaction that we call chemical reaction that we call hydrolysis. Note, saliva is a digestive juice where salivary amylase is an enzyme inside the juice. And salivary amylase is an enzyme which later we know and they are protein. It's a kind of specialized protein that can help speed up, catalyze the breakdown of starch into maltose. There's another note that amylase cannot digest starch into glucose directly. You can imagine the amylase being kind of eater that bite two glucose at one time, and it cannot bite one glucose. So every time it bites, two glucose, that is a maltose molecule, comes up. Another part of the alimentary canal, which is I would like to talk about, is the stomach. In the stomach, there will be mechanical digestion by contraction of the stomach wall. The contraction of the stomach wall is dynamically termed as churning. The stomach wall secretes gastric juice, which contains protease, which is an enzyme to break down, actually to speed up the breakdown of protein into peptides, to be precise, and change milk protein into insoluble one. Personally, I don't really agree with this because when protein breaks down, it should change from being insoluble to become soluble. So breaking down protein should not be the reason for the milk protein becoming insoluble. So there should be other reason for milk protein to become insoluble. We have a term called coagulation, which you might be interested into looking into up in the web, the Google web, to find out how milk coagulate. But that is not the main issue to discuss right now. Hydrochloric acid is also present to kill microorganisms in food and provide an optimum pH. That is, provide an acidic medium with a pH 2, use hydrochloric, the pH of hydrochloric acid, very acidic, for protease. You might question why the hydrochloric acid being pH 2 very acidic and corrosive. It won't hurt, it won't damage the stomach wall. And that is because there's a mucus on the stomach wall to prevent the stomach wall from being digested by protease or harmed by hydrochloric acid. The next structure along the alimentary canal that I would like to discuss is the duodenum of the small intestine. The small intestine consists of duodenum and ileum, and the first part is the duodenum, which is a very short, consists of 3 digestive juices. There are 3 digestive juices found in duodenum. The first juice is called bile, the second juice is called pancreatic juice, the third juice is called intestinal juice. For bile, it contains bile salt, which can emulsify lipid, change the lipid into small oil droplets so as to increase the surface area for lipase, which is an enzyme to speed up the breakdown, to catalyze the breakdown of lipid into glycerol and fatty acid. There are also sodium hydrogen carbonate, which is an alkaline, a strong alkaline with about pH 11 to neutralize the acid from stomach. And this happens to provide an alkalinic or alkaline or an optimum pH, which is an alkaline medium for the enzymes in duodenum. Enzymes you will learn later, different kinds of enzymes have different optimum pH. Pancreas produces pancreatic juice, which contains pancreatic amylase to digest starch into maltose. Pancreas also contains lipase. The pancreatic juice also contains lipase, the enzyme, to digest lipid into fatty acid and glycerol, as I have just said. Pancreatic juice also contains protease to digest protein into amino acid and peptides. The pancreatic juice also have a strong alkaline that is the sodium hydrogen carbonate, to neutralize the acid from stomach and consequently provide an alkaline medium for enzymes in duodenum to work properly. That is to digest the food properly. Intestinal wall being the third structure to produce the third digestive juice. And the digestive juice contains different carbohydrates. Well, carbohydrates, examples such as maltase. You remember amylase digests starch into maltase, but it has to be glucose, small, so that it can be small enough to be absorbed into blood. So there is another enzyme called maltase, which we group together this maltase or sucrase, which digests sucrose into fructose and glucose. These together, they are called carbohydrates. They digest various sugars such as sucrose and maltose into monosaccharides, such as glucose and fructose or galactose. The intestinal juice also has proteases. To digest peptides into amino acids. But I would like to tell you that these information are a little bit outdated and not really correct. These information are given here for easy understanding in the early years, such as the HKCEE years, but now in HKDSE years, we are modifying, we are uh, making this information a little bit more precise, that the intestinal juice does not really contain these carbohydrates and proteases. In fact, these carbohydrates and proteases are situated on the wall of the small intestine, on the cell membrane, on the proteins of the cell in the cell membranes of the small intestine, of the epithelium of the small intestine. Please go to page 18 of your book, chapter 6, to clarify, to identify all this information. And I would not reiterate here in these notes again. A summary of digestion. For starch, saliva is responsible for digesting starch into maltose. Pancreatic juice is also responsible for digesting starch into maltose. Intestinal juice, so better change it to this word. Intestinal juice is responsible for giving a good environment, an optimal pH for the digestion of starch, for the digestion of maltose into monosaccharides, that is the glucose. For the digestion of protein, gastric juice is responsible for digesting protein into peptides. Pancreatic juice is responsible for digesting peptides into amino acids or protein into peptides. For intestinal juice is responsible for producing a good environment and optimum pH for enzymes on the wall of the intestinal, the small intestine, wall of the small intestine, to digest peptides into amino acids, which you should refer to page 18 of your book, chapter 6. For lipid, there are bile, which physically digests oil or the fats into oil droplets. And pancreatic juice, which are responsible for digesting fat into fatty acid and glycerol. Movement of food along the alimentary canal. We have a process called peristalsis. Peristalsis itself, I would like to state here first, is also a kind of mechanical digestion, because it helps to break food up into smaller pieces. Here, peristalsis is a depiction of what the nature is of peristalsis. This is a wave of contraction and relaxation of circular muscle of canal wall. These are circular muscle. They contract to push the bolus forward, and this circular muscle relax to allow the bolus to pass through. Now we move on to absorption. Structural adaptation of small intestine for absorption. The small intestine is long. It provides a large surface area and allow more time for complete absorption of food. It is highly folded. The inner surface is highly folded, which possesses a lots of numerous that means a lot of villi, to increase the surface area for absorption of digested food. That is the fatty acid, glycerol, the amino acid monosaccharides such as glucose, fructose, and galactose. These are examples of absorbable form of food broken down into the smallest possible form of protein, fats, and carbohydrates. Structural adaptation of the villus. This is the villus, and structural adaptation of the villus is here. It is finger-like. It provides a large surface area to volume ratio for food absorption. It is thin-walled to provide a short diffusion distance for diffusion, so that it is quicker in absorption. It is richly supplied with blood capillary and also lacteal to transport the absorbed product away rapidly so as to maintain a high, better say, not high, a steep, see, this is a better way of expressing, steep concentration gradient, and therefore diffusion is faster. Large number of microvilli on the villi, that is, these are the microvilli on the villi. Which is this one. It's literally some extension of the cell membrane to make these microvilli. Also increase the surface area to volume ratio of the cell for food absorption. These cells on the surface of the villi are called epithelial cell. But these epithelial cells are so differentiated to have microvilli on the top for efficient absorption of food. Absorption of digested product. Glucose, amino acid, and mineral salts are absorbed together with water, of course. They are dissolved in water and move inside with water into the blood capillaries. Fatty acid and glycerol, first taken up by the epithelial cell that I've just talked about in the last page, and are recombined into oil droplets inside the cell before they are moved out of the cell and transferred to the lacteal. Absorption of other food substances, mineral salts and water-soluble vitamins, because they are water-soluble, are absorbed into the blood capillary. Fat-soluble vitamins, they will go together with the fats and enter the lacteal. Absorption of water, most of the water is absorbed in the small intestine because they are very long, so most of the water is absorbed there. Actually, water is starting to be absorbed in the mouth, but since mouth is shorter compared with the long small intestine, so most of the water is absorbed in the small intestine. The absorption of digested food in the blood increases the water potential of the content of the gut, which you will learn in chapter 4. In chapter 3, the water potential. As a result, water is drawn into the blood by osmosis, which you will also learn in chapter 3. Part of the remaining water is absorbed in the large intestine, that is the colon, together with mineral salts. Because the water, mineral salts dissolves in water. so it is absorbed together with the water. The next process is called assimilation. It is the process by which the body cells take up and use absorbed nutrients or food substances. The route is being shown below. From the intestine to different kinds of tissue. For glucose and amino acid, first capillaries in the villi, and then the hepatic portal vein. The portal vein happens to be a blood vessel between two organs, and there is basically only one portal vein that we need to know in DSE, the hepatic portal vein, and then which shunt blood from which allow the blood to go from the small intestine to the liver, and then from the liver it moves out through the hepatic vein, and then to the largest vein of all, called the vena cava, and then to the heart, and then pump out of the heart through the pulmonary artery. Pulmonary is a word that means the lungs, which this artery goes out of the heart and to the lungs. And then after the lung, the blood moves out through the pulmonary vein, and it goes back to the heart. So all arteries move blood out of the heart, and all veins move blood towards the heart. Literally, for blood that moves towards the heart, the blood vessel is called the vein. For blood that goes away from the heart, the blood vessel which carry them are called the artery, which we will learn a little bit more in chapter 8, the circulatory system. But we'll move on right now. The heart, and then out of the heart, the largest artery of all, the aorta, and then artery and various organs, different organ, and then reaching different kinds of tissue cells. For fatty acid and glycerol, there is a little bit of a change in the route. First, recombine to form lipid molecules at the epithelial cell on the villi, and move into the lacteal of the villi, and then move into the lymph vessels, which is part of the lymphatic system, and then move to veins, and then vena cava, and then heart, basically the same route now, pulmonary artery, lungs, pulmonary vein, heart, aorta, arteries, and various organs reaching the tissue cells. Now, the fate of all these absorbed nutrients would be like this. Nutrients, which we put examples, would be glucose, amino acid, and carbohydrates. I particularly put glucose and not carbohydrates for simplicity's sake, so that to make things easier for understanding. Fate would be excess glucose is converted into glycogen and stored in liver and muscle. Glucose is oxidized. In oxidation, these glucose are broken down. For oxidation, you will learn a little bit more from chemistry lesson, or from your biology teacher who will particularly focus on certain parts of chemistry to teach you in the case when you don't have an elective called chemistry in your subject. But the fate of glucose would be broken down to release energy in all tissue. These energy are in form of ATP, adenosine triphosphate. A little bit of elaboration here is that the ATP, this is A, the adenosine, which what we call a special thing, a special part called base, has 3 phosphate group, and when the phosphate is broken down into 2 phosphate ADP, adenosine diphosphate, then the energy will be released for doing anything such as muscle contraction. The amino acid, the fate would be like this. Excess, too much amino acid, will cannot be stored. It will be broken down in the liver to form urea for excretion. The non-nitrogenous part of the amino acid can be converted into glycogen. Of course, the nitrogenous part would be excreted in the form of urea in urine. The amino acid will be used to synthesize protein for growth and repair of tissue. For lipid, excess lipid can be stored. It is stored under the skin or around organs such as the liver. It is oxidized to release energy in liver. It is also used to form cell membranes, which you later on will learn that cell membrane consists of mostly lipid and protein. The role of liver in assimilation would be the regulation of blood glucose level. A little bit of elaboration here is that the pancreas is responsible for releasing two hormones. One is called the insulin, which we know as 胰岛素, and the other is glucagon, which has the opposite effect of insulin. And these two insulin will stimulate the liver to change glucose. This insulin will stimulate the liver to change glucose into glycogen, where glucagon will stimulate the liver to change glycogen back to glucose, and the pancreas will detect the blood glucose level in the blood and then decide whether to release more insulin or more glucagon. And when the glucagon and insulin, these are hormones, are released into blood, they travel to the target organ called liver, where the liver cell will be stimulated by these hormones to convert glucose to glycogen or glycogen to glucose, which you will learn later on in later chapters. But we have a little bit of glimpse here of the information of how regulation of blood glucose level is done by the liver. It is also responsible for storage of glycogen, iron, and fat-soluble vitamins, because the liver is also a place to store fat. So anything like vitamins that are soluble in fat can also be a place where it can be stored. For example, vitamin A and vitamin D, and not vitamin C, because it is water soluble. Breakdown of excess amino acid is also done in liver. The process is called deamination, which I have just talked a little bit about in the previous page, where excess amino acid cannot be stored and will be broken down in into the liver, and the non-nitrogenous part, this is the carbohydrate part, will be changed to glycogen, and the nitrogenous part, that is the amino group, will change and become urea and be excreted into in the urine. So the role of liver is for breaking down of too much amino acid. Egestion. It is the process of removing undigested materials. So these 5 processes: ingestion, digestion, absorption, assimilation, and egestion, are not necessarily in order. So egestion happens to begin after digestion, and it also happens after absorption, to remove undigested and unabsorbed nutrients from the alimentary canal. So we can basically write the order of these 5 processes like this. First with ingestion, then digestion, and for digestion it can go to the process absorption or be digested for undigested materials. As for absorption, it can go towards assimilation, and then or unabsorbed materials, it will go towards digestion. Feces are formed when water in the waste materials is absorbed. It should be changed to is here. Is absorbed by the large intestine or colon. They are temporarily stored in the rectum and expelled through the anus. So that is all I would like to tell you in chapter 6. For other information, please read through the book, and I would like you to do these MCs and the long questions or structured questions for me to mark and later on discuss with you for any mistakes that you have made. So start working.